I'll jump straight in. So, today I'm going to talk about HIV epidemiology, and I wanted to start with introducing you to some of the methods that I normally use in my analysis. Basically, we obtain sequences and then we use phylogenetics to reconstruct pretty trees that represent how the sequences relate to each other, as in figure A. We then use molecular clock theory that assumes that organisms evolve over time with a relatively constant rate to translate these genetic trees onto time trees. That helps us estimate when certain events happen along the tree. For example, certain mutations or transmission events, or what we suspect to be transmission events. If we have even more information, as in figure D, for example, where were the samples taken or which risk group were they taken from, we can then estimate how the virus moves between these locations and between these groups. As mentioned, I do a lot of work on HIV. And today specifically, I will highlight the work that we've been doing in Ukraine in the last pretty much 10 years. I used to start all my talks with a map of Ukraine, which no one needs these days. But what people don't often know is that Ukraine actually has one of the largest HIV epidemics in Europe and has been battling it with successful prevention strategies for the last 25 years. But unfortunately, back in 2013, 2014, when we were just starting to get the HIV epidemic under control, the number of annual new infections was starting to drop. The war started in Ukraine. It didn't start 2 years ago, it started 10 years ago, first with the protests on the main streets of Kiev, that then escalated in the annexation of Crimea and the occupation of the eastern parts of the country. At the time, I was a PhD student working with my supervisors, trying to understand how will that affect the HIV epidemic in the country. And one of my mentors, who's a sociologist by training, has developed this framework that we call big events framework that describes how big events, disruptive events such as military conflicts, environmental disasters, economical conflicts, can disrupt the current norms, physical norms, social norms, cultural norms, and how that will have certain epidemiological effects. Some of them will be early or immediate epidemiological outcomes. For example, if a hospital is bombed and people cannot access treatment there anymore, people will be out of care, they will have a higher viral load, transmissions will happen. Some of them will be later epidemiological outcomes. For example, when in situations of protracted conflict, the infrastructure goes down. We heard a lot from Luke about that. And that results in people regularly not having access to treatment and, again, onward HIV transmission. A few years back, I used data from surveillance, from the drug resistance surveillance for HIV, to reconstruct HIV phylogeny for Ukrainian data and apply phylogeographical analysis to study how does the virus move between different parts of the country. What we were able to show was that the virus moves from the occupied areas from the east into the rest of the country. And the number of viral migration events is actually correlated with the number of people who are moving. However, this was surveillance data. We didn't have any information about who these people are, how to reach them, whether they're staying in care once they reach new places of residence. So what we decided to do is to go and ask these people about their experiences. So in 2020, during the pandemic, together with amazing colleagues present here in this room, we went to Odessa in Ukraine, and we interviewed a small sample, 164 people who inject drugs, who are at risk or living with HIV, who have been displaced by the war. First of all, what we noticed is that a much higher proportion of them are staying out of care. In the figure in the bottom right corner, you can see that about the same proportion of people as any other person living with HIV in the country knew about their status if they were living with HIV, but only about 40% of them were receiving care, and also a very small proportion of them were actually virally suppressed. So we've collected samples from these people, and we also asked them about their migration experiences. We asked them, when did you come to Odessa? Where were you before? And then with a super team, we went to Odessa to sequence some of the viruses. We used Nanopore because there was no other pretty much sequencing platform available in the area we worked with. And we tried to make the conditions that we worked with as close to field conditions as possible, and not rely on the infrastructure of the hospital, because we thought, okay, what if the hospital is not there next time we're here? So, what's going to happen? And we had the best time. As a person trained in phylogenetics and in epidemiology, Luke was the one who taught me how to hold the pipette for the first time in my life. To be in the field and do the sequencing in the lab, in our Airbnb, in a hotel, on a drive back to the hotel in the car, on a drive to the airport because we're being kicked out of the Airbnb and it's time to check out, but the run is still going. It was a lot of fun. And importantly, we were there to train people. All these people in the lab, they always wanted to do sequencing. This is a diagnostic lab, but they were like, okay, we don't have that capacity. And we were like, okay, we can't use this for diagnostics, but we can do it for research. So we can train you how to use that, and you can watch us do it. And this was a lot of fun and very engaging work. Finally, we got to the sequences, and we were able to estimate and infer some trees. Why do we even do that? Because when we test people and we know that they are living with HIV, people can be infected with HIV for 10 years. We don't know, were they infected before they got displaced, after they got displaced, when did it happen? And this is a crucial question for prevention in this setting. So what we do, we estimate a tree and we try to find transmission clusters, people who are genetically sharing viruses that are very similar to each other. What we then do, we apply molecular clock theory and we try to estimate the time of when did this cluster originate, potentially telling us when did this transmission event happen. And we saw that out of 8 clusters that we found, 2 of them, as you can see in the posterior density distribution at the bottom, happened after the start of the war. We did a similar analysis with hepatitis C, and in the same manner, we found some clusters, we estimated the time of their origination, and we also mapped when did people who are in these clusters move to their new place of residence. So the purple dots on this figure on the left is the time that the last person in the cluster actually reached the Odessa region. So for the 2 clusters that did originate after 2014, we estimated the interval of when is the plausible origination of that cluster, then mapped when did people in this cluster reach Odessa. And what we saw was that in the cases when people did get infected after displacement, they got infected within the first year upon arrival. In situations of protracted conflict, which are the majority of situations in the world, where people are staying for 10, 20 years in their new place of residence, it is crucial to understand that when infections happen, they happen early on, so prevention needs to be administered immediately and not after, 5 years. Because we were already there, it was the middle of the pandemic, it was 2021 when we came back and did some more sequencing. Delta was rampaging around the world, and Ukraine is one of the places where we don't have sequencing capacity. It was a year and a half into the pandemic, and there were 100 sequences on GSAID from Ukraine, while there were hundreds and thousands of sequences from other places. So we decided to use the technology that we had at hand to sequence some more. We sequenced 100 more, so we doubled the number of sequences. We were very proud. All of them were Delta sequences. And what we've done, we then placed them on the global Delta tree, and we asked ourselves, okay, so what happens here? Like, why are all of the sequences being introduced from all over the world, Ukraine is not that well connected. And what we realized when we looked at some of the tourist data, we realized that Ukraine in summer 2021 had more tourists than before the pandemic. What happened? All the other European countries had some sort of travel restrictions, travel bans, testing, and Ukraine was free for all. So it became a popping tourist destination, which resulted in a bunch of introductions of the virus into the country. I also wanted to highlight a project that we're just starting in Ukraine, and it's going to focus even more on the combination of the social network analysis and phylogenetic analysis and trying to relate the changes in the number of social ties that happen as a result of a displacement and disruption of the social network, and how that relates in the creation of genetic ties, really linking the social behavioral and the genetic epidemiology. With that, I would like to highlight all the amazing collaborators who's made this possible, and thank you for your attention.